A very good evening to all the participants. Knowledge is like a garden. If it is not cultivated, it cannot be harvested. I am Mahima Dikshit, the co-organizing secretary of the one-month international training on precision agriculture organized by CSGM University Kanpur and AgriMeet Foundation in collaboration with ICRISET Hyderabad welcomes you all to day 18 of the international training on precision agriculture. The title for today's training is Precision Phenotyping for Abiotic Stress Management. To share his views and knowledge, today we have our eminent speaker, Dr. Shivashakti Kalimurthy with us. Let me take the honor to introduce our eminent speaker to the participants. Dr. Shivashakti Kalimurthy is an associate scientist of sorghum and pearl millet diversity panels for water use and agronomic related traits under well-watered and water-stressed conditions using lysimetric facility. Previously, he was a research fellow working on phenotyping of stay green chickpea for water use in agronomical traits. During his PhD, he focused on contribution of water-saving traits for drought adaptation in chickpea through physiological, molecular, and genetic approaches. He has a good understanding and working experience with scan lysimeter and field experiments. He has gotten knowledge on plant hydraulics by using pharmacological approaches by handling various physio instruments such as Spolander pressure chamber, IR camera, septometer, etc. He has supervised 5 master students and 3 interns. He has published 10 research articles and 2 book chapters. Further research interests include assessment of grain nutritional content and genetic enhancement of carotenoids content in different stay green legumes like chickpea, cowpea, common bean, lima bean for nutritional security, in addition to assessment of genetic variability in fodder and grain quality parameters in sorghum and pearl millet diversity panels. I am honored and pleased to welcome you to day 18 of Precision Agriculture Training. We welcome you and we would like you to share your knowledge with us.
Yeah, thank you. Thank you so much for your introductions. Yeah, it's really great. So I had opportunity to present this wider audience. So I hope my presentation will really helpful. So just I have one question. How can I shall I share my screen or how I can proceed with this?
Please share your screen, sir.
Okay, thank you. I hope everybody can see my slides.
Yes, sir. Yes, sir.
Okay, yes, shall I start?
Yes, please.
Yes, okay. Thank you everyone. Thank you everyone. So good evening everybody. So myself, I'm Shivashakti Kalimurthy. So in a postdoctoral fellow, so working in a crop physiology team at ICRISET. So my work mainly focus on this abiotic stress adaptations. So today I'm going to talk about the precision phenotyping for abiotic stress adaptations. These presentations, we mainly focus on abiotic stresses and its impact and component traits for abiotic stress adaptations, as well as I'm going to cover this high throughput phenotyping platform such as that exists in ICRISET, such as scan and lysimetric facility, and some of the relevant case studies for drought stress adaptations and heat stress adaptations, as well as salinity. Also, finally, I'm going to talk about a little bit about low soil phosphorus stress adaptations as well. We look at various abiotic stress such as drought, heat, salinity, and low soil fertility. All these stresses significantly impact plant growth and development. These stresses ultimately decrease the crop productivity on both grain, legumes, as well as cereals. Therefore, it is very important to understanding their effect is crucial for developing climate resilient crop varieties. When we look at these effects in different abiotic stresses on crop yield, it is very clear from the slide, abiotic stresses have substantial impact compared to the biotic stresses. If you see the maybe yellow bars, so those are in the abiotic stress yield losses. Almost all the crops, all the major cereal and legume crop, both 60 to 70% yield losses. But when we compare to the biotic stresses, that's there in the bottom as a green, around averagely 15 to 20% yield losses. So it's very clear evident that all our major agricultural crops, that abiotic is a major concern for yield losses. Yeah, when we look at very closer, so climate change will bring both positive and negative impacts on agriculture. Rising temperatures may extend growing seasons and also increase crop yield only some regions, but extreme temperature events that can lead to heat and heat stress as well as the crop failure. Changes in rainfall pattern and amounts of the rain and timing of the rain, that can all this can result in drought and flood and soil erosions. This can be, we can very frequently we are seeing. These projected impacts highlights the need of research for abiotic stress adaptation. Therefore, it is important to enhance our understanding for target agroecology through environmental characterizations. So yesterday, our colleagues explained or provided great details about environmental characterizations. So I'm not going to talk about in details, but very briefly, I'm saying that I'm going to talk. This environmental characterizations, this particular process, this particular things really enable us to identify and address crop production constraints, due to the climate change events. As a part of environmental characterizations, our approach involves several steps. The first step, we identify target environment, and next step, we try to understand abiotic constraints in the given environment through a lot of crop modeling tools. And later, we proceed for designing the crop that suit for target agroecology. Also, we try to understand the crop management practices. This will lead to for improving the crop yields. By following this process, we aim to adaptation of crop variety in particular target population environment. That's very, very important. So it is not for all crop varieties, not for adapt for all the environments. So our focus mainly the target agroecology based. While yield is a primary focus for everyone, so everyone wants yield. So, but we need to understand one thing. So it is very important to recognize grain yield is influenced by several component traits. Each of these component traits hold its own significance and contributes for the yield formation, particularly when its interaction with the environment. Therefore, it is very crucial to shift our attention towards understanding and improving these component traits rather than solely focusing on overall grain yield improvement. For example, in the context of drought, we specifically emphasize importance of water use related traits as a play direct role in drought adaptation. Now, let's take a closer look at the specific component traits that focus on our current research. These traits encompasses various aspects in relation to water utilization and drought adaptation. They include limited transpiration rate under high operative demand. That is simply we called causes by atmospheric drought. And second one for this soil water conservations, that will be soil drought due to the deficit of water. And third one for the canopy development. So, and fourth one for this root hydraulic conductivity. And fifth one for water extraction at key stages of plant growth. And the last one for the transpiration efficiency. Yeah, here I would like to re-emphasize that any trait or allele that can confer the drought tolerance, so we need to just design the right drought scenarios. So I like this paper very much. So this paper clearly suggests that all the traits has one value within specific environments. We need to select right trait and test target agroecology. Yeah, if you like to explore trait values or the expression of trait in a given environment, yeah, please use crop modeling tools. It will always allow us to explore your traits in a more locations, in a different combinations of treatments. That is the beauty of this crop modeling tools. Yeah, if possible, please read this paper. You will get a lot of information about trait value in a given environment. Yeah, next, we just focus on the specific water saving traits that I listed before. The first one, we're going to see the limited transpiration rate under high operative demand. For limited transpiration, here is one of the example I given the pearl millet. And the x-axis, we have a vapor pressure deficit, which is represents simply atmospheric evaporative demand. As VPD increases, as the vapor pressure demand increases, the drought sensitive genotypes in a red continuously loses the water. When drought adapted genotypes in green, that begins restrict water losses around the 2 kilopascals. Yeah, these figures clearly illustrate that how different genotype responds varying evaporative demand. Really, these differences in transpiration directly linked to soil moisture conservation that eventually contribute to crop adaptation to drought. The next trait, soil water conservations. Let's focus soil water conservations. Here I'm giving one of the example of chickpea. As soil progressively dries out, that's a regulated progressive soil drying, we observe that there is a substantial genotypic variation in soil moisture utilization in different kind of genotypes. As I mentioned, this chickpea example, so drought sensitive genotypes is mostly, utilize soil moisture rapidly, quickly, than drought adapted genotypes. So these differences in soil moisture utilization that clearly reflected on crop adaptation to drought or the water limited environments. So this is a very clear-cut evidence that soil moisture conservation is really important for drought adaptation. The next one for the canopy development. For the canopy development traits, so one of the case study in chickpea, during heat stress, it is found that heat adapted genotype of chickpea have better canopy development than heat sensitive lines. These differences in canopy development may lead to differences in soil moisture utilization that eventually resulted in crop adaptation to abiotic stresses. The next trait for the root hydraulics or water transport pathways. Yeah, water transport pathways, I would like to give brief description. So in the root water uptake process, there is a two kind of water transport movements, one for this radial water flow movement and another one for the axial water flow movement. For the radial water transport, there are two major pathways involved. One is apoplast pathways, another one is symplast pathways. The symplast pathways majorly water flow through occurs through plasma tesmata cell to cell. This water is moving or channeling towards the xylem. Our apoplast pathways, that's intracellular spaces. Through that, water is moving through the xylem. From xylem to water moves through the axial water transport through all the plant tissues. That's all like a little bit of basics. Yeah, we also found that even the channeling of water transport pathways in a different kind of material or different adapted lines, they have really clear-cut difference. Okay, now we are looking at this root hydraulics point of view. We found substantial differences between drought adapted and drought sensitive genotype in terms of dependence of various water transport pathways, such as the symplastic as well as apoplast pathways. Here is one of the example of pearl millet. Drought adapted genotype in a green uses less symplastic water transport pathway and more apoplast pathways. These differences, found to correlate to drought adaptation in a particular crops in a given environment, in a given conditions. The next trait, I will move for this water extraction at key stages of plant growth. Water extraction at key stages of plant growth is very, very important. Here on y-axis, we see water used as a function of time after panicle emergence. Irrigation is stopped at time of panicle emergence. Under this circumstance, drought-sensitive pearl millet genotype in red color, the red line, extract more water before flowering, and so it does not have enough water after flowering time or this grain filling time. And contrary, the drought adapted genotype in the green color dotted lines that extract less water before flowering. So whatever water saved during vegetative to that flowering time, so that all the water, saved water can be utilized during after flowering during the seed filling time at the reproduction stages. These differences in water extraction is clearly reflected in a grain filling. Here you see that this adapted genotype, they're able to fill the grains efficiently than sensitive ones. So this is very important for water extractions in terms of drought adaptation point of view. The next slide, I'm going to talk about this transpiration efficiency. Transpiration efficiency is a measure of plant ability to use water for photosynthesis while minimizing water loss through plant transpiration. It indicates potential to produce greater biomass while less water. Recently, we evaluated TE of sorghum diversity panel, and we found that substantial genotypic variation in TE. In the graph, the cultivars are represented in the yellow bars and diversity panels are represented in blue bars. Compared to cultivars, some of the germplasm lines had higher TE values, which could be used as a donor in a breeding program to develop cultivars that are adapted to water limited environments such as drought conditions. So far we have seen about the several specific water saving traits that has different impacts on grain filling and crop productions. But still I see that phenotyping still is a major bottleneck for the crop improvement programs. Even though we have a lot of traits, but the phenotyping still not really so easy. So our current field-based phenotypings often labor intensive, time consuming, and lacks in, we don't have much precision data sets. Additionally, many phenotyping methods are destructive nature, limiting ability to study the plants over time. We cannot see the plant responses over the time. And furthermore, spatial and temporal flexibility required for large-scale phenotyping in testing of large number of entries really difficult to achieve. The need of high throughput phenotyping platforms. So why we need, if I look at that, The first point, it's require less manual labors due to higher automations. And second points, if I look, it enables fast and non-destructive based phenotyping and provide precise and accurate data. Also offer minimal restriction in terms of phase and time. And it allows for easy phenotyping for entire population. Also facilitates a dynamic phenotyping. That's really very important, that leading to increased selection efficiency and accelerate the genetic gain in the breeding programs. In the field of crop phenotyping, we have high throughput, highly automated phenotyping platform, such as called LysiScan. So I'm going to often use this terminology, LysiScan. That means that high throughput phenotyping platforms. This is an innovative system that utilizes novel 3D scanning technique to constantly monitor the plant growth and development in a non-destructively on daily basis. In addition, this technology utilizes scanner to plant concept to enhance speeding imaging. Faster imaging this technology can produce. This is a world-class unique facility that available at ICRISAT to support crop improvement programs as well as much more. Within this platform, we have capacity to scan 5,000 microplots in a span of 2 hours. By using this high throughput phenotyping facility as a LysiScan, we were able to effectively capture the traits that are associated with the canopy development. For example, the structural traits, the canopy development, here we see that digital biomass and 3D leaf area, projected leaf area, plant height, leaf angle, all these things are comes under the canopy development related traits, and functional traits such as this plant transpiration and water use efficiency. All these kind of things we can estimate. These traits are really crucial for adapting stress conditions such as drought, heat, salinity. These facilitators really support us to screen all these abiotic stresses. Next to LysiScan, so we have another facility that is called semi-automated phenotyping platform. That's called as a lysimetry facility. Beauty of the system is it can be used for measurements of both agronomy as well as water use related traits, which is impossible in the field environment. It consists of PVC tubes such as 1 meters, 2 meters length with a diameter of 20 centimeters. So we will fill this cylinders in this soil. The arrangement of cylinders, the way the field density. So we are our aim is this data we're going to compare with this field. So we always maintain this field density. If it is legumes, 28 to 32 plants per meter square. If it is cereals, around 16 plants per meter square. Accordingly, the cylinders will be arranged. These PVC tubes can be lifted with help of hook and pulley chain that attach with the load cell that can help in measuring the weight of cylinders. So through weekly weighings, we can exactly quantify the amount of water extraction that occurs each crop varieties or each plants. Furthermore, this system enables us to measuring the standard agronomic traits as well as agronomic packages. Similar to the field settings. The system protected by rainout shelter. These particular things really enable us to precise management of drought experiments. So we don't need to worry about for the rain and other things, because this shelter really taking care and closing are protected from the rain. Here is a little bit of closer image of these lysimeters. So we have capacity of 4,500 lysimeters. We have different soil types in that. We have wetty soil, alphy soil, loamy soil, sandy loam, sandy, different soil types. We can also explore the effect of soil on plant growth and development. By using this facility, we can precisely quantify genotype by environment, by management interaction, all G by E by M by interactions, all we can explore. Yeah, apart from water use components, we just talked about this lysimeter facility for only for water use components, some of the agronomic components. Apart from these components traits, we can also phenotype root morphological traits that are linked for crop adaptation to drought using this lysimetric facility. Yeah, this facility recently earlier we have screened for large diversity panels of sorghum germplasm set for root morphological traits, root length, density, root surface area, root volume. These are all some of the morphological traits that contributes for the drought adaptation. Using these systems, these root traits were phenotyped and also introgressed into the elite cultivars. So there are a lot of mapping populations were developed. Lot of advanced materials were developed by introgressing these root QTLs. Recently, 3 varieties were released at IRI in this ICRISAT root introgression lines are released in collaboration with the NAS partners. So next, so everybody have always have idea. Okay, so whether your water use related traits or the high throughput phenotyping platforms, whether that traits can be linked or that traits have any relevance to the agronomic practices, whether they have any trait correlation with agronomic traits. I would say really yes. Water use related traits that we phenotyped from these lysimeters were found to very close association with yield. In the slides, this association between grain yield and T was shown in pearl millet and sorghum diversity panels. So I could see that transmission efficiency and grain yield very tight associations. So from this, I will say that T really a viable trait for the breeding programs to accelerate the genetic gains. So here, a very clear-cut example: if breeder breed for high TE lines, surely they will get better yield in a water-limited environments. Here is another example from grain legumes such as cowpea, chickpea, common bean, lima bean that were evaluated in the lysimeter facility for water use and agronomic-related traits under well water and water stress conditions. So under well water conditions, seed yield is mainly driven by biomass. Under water stress conditions, biomass influenced by transmission efficiency. This lysimeter facility has helped us to understand importance of water use related traits on crop production related traits. This knowledge can be used for designing specific breeding product in a particular environments or target agroecology. Next, we will going to see on some of the case studies for drought, heat, salinity. So in this ones, I'm going to talk about this case study on drought stress adaptations. Here, we used in a chickpea RIL populations. This is one of the case study of chickpea. In this case study, we used a chickpea RIL population to phenotype and map traits related to water use, canopy development related traits. It took 7 seasons and 5 locations for our colleagues to phenotype classically for agronomy related traits, as well as some of the root related traits, to map genomic regions that are associated with the chickpea yield. However, we phenotyped plant vigor and water related traits in just a matter of 3 to 4 weeks using these high throughput phenotyping platforms, and we were able to map exact genomic regions that were previously reported. So in chickpea, they will say one favorite thing, QTL hotspot region on chromosome 4. That's everybody interested and looking at this root QTL introgression lines. So that particular QTL hotspot regions, our canopy development traits are exactly co-localizations that happen. So the data derived from these high throughput phenotyping platforms are precise and more accuracy. That's what we land up with similar co-localizations and adaptation for drought related traits. Here is another case study on PCMP. So in this case study, we used PCMP breeding material that are contrasting duration. Some of the lines are the short duration, some of the lines are long duration varieties. In the short duration plant type, on the right, the seed yield predominantly determined by shoot biomass under drought stress conditions. By contrast, long duration type, in the graph, if you see the left side, the RS index is a major driver for seed yield. So BGMP breeding program can use this knowledge to develop cultivars under water limited environments according to the duration of the materials. They can really focus on different duration materials, different target traits that can be clearly visualized here. The next case study, I'm going to talk about the heat stress adaptation. Yeah, in this heat study, in this heat case study, here we are going to use this wheat non-population parental lines, that material derived from CIMMET non-population parental lines. So when we screen this wheat material for heat stress using our high throughput phenotyping platforms, we found heat adapted lines shown in blue lines that have better canopy development than the heat sensitive lines in this orange, the bottom ones. This is very important for canopy establishment during heat stress. Without canopy establishment, we cannot aim for any other agronomic things. This facility is able to catch up differences in plant growth very efficiently, which is not possible in the field phenotyping methods. To continue that, the wheats are evaluated 2 different seasons. One is some kind of mild heat stress, another one is severe heat stress. So here, all these box plots are showing those blue ones are this mild heat stress, and these red ones are the severe heat stress. There is a very clear-cut evidence that heat stress clearly affects the phenology that will eventually reduce its yield and other component traits. Also, this also reduces biomass, plant height, number of protected tillers, all those things, it's severely reducing that. So the beauty of this, so all these things can be very, very well captured in this high throughput phenotyping platforms. Sometimes we cannot be able to capture this field phase to be phenotyping, especially this dynamics of phenotyping things. Next heat case study is in chickpea. They're similar to wheat. We have tested around 1,000 accessions in this high throughput phenotyping platform for the chickpea mission project. We have tested 2 different seasons, one for the normal rabi seasons, another one for the summer seasons. We could see very clear-cut differences in plant growth and development, around 62% reduction in canopy development or this leaf area due to the heat stress. So yearly vigor or biomass accumulations really crucial for heat stress adaptation in chickpea. So this is very clearly evident that the chickpea very sensitive for the heat stress that can be easily captured by our systems. The next case study, I will move on for the salinity stress adaptations. For the salinity case study, so I'm used for the chickpea. So here we used around 1,000 accessions that were screened in our high throughput phenotyping platforms. These materials are part of chickpea mission project. We screened these materials for the salinity or the saline condition as well as the normal conditions. To see, here we clearly see that salinity stress reduce the canopy development around 45% compared to the non-saline one, the control plots where we receive the normal water. Under salinity, the yearly plant biomass accumulation is really, really important for crop adaptation to salinity. So all these things, our facility nicely captured. For another case study on salinity in pearl millet, here we see that the plant transpirations were measured in hourly basis to look at that diurnal pattern of plant transpiration response. In a day, we just progressively monitored the plant transpiration. Some point, we added salt solutions. After that, we could clearly see that around midday, there is a clear dropping of a clear reduction of transpiration due to the salinity stress that can be very nicely captured with help of our systems. So these differences, all these differences will link for crop adaptations. For the next case study, I will move on for the low soil phosphorus stress adaptation. That also very important compared to, that also very important in addition with this abiotic stresses. So yes, especially for fertilizer inputs, phosphorus, nitrogens, they are really key for agriculture production. Yeah, in this low soil phosphorus stress adaptation case study, we have tested, we have used for this foxtail millet crop. We have tested the foxtail millet mini-core germplasm lines in this high throughput phenotyping platforms. We have planted this material in the low soil phosphorus conditions where we mimic less soil. There is very less soil phosphorus. Another set of plants we given optimal phosphorus and the NPK optimal phosphorus dose. In this low phosphorus one, we have provided sufficient nitrogen and also the like potassium. Only deficient is phosphorus. So these lines were screened our high throughput phenotypic platform for canopy development related traits. Here we clearly see that those blue lines, the low soil phosphorus, under low soil phosphorus conditions, they have very reduced canopy development compared to the optimum one that are in orange ones. The optimum ones, they have a better canopy development. They have higher canopy development. So we also found that a substantial variability in number of tiller formation under low soil phosphorus conditions. The genotypes that are adapted to low soil phosphorus conditions that produce more tillers, which is critical or key for better input or resource use efficiency in the poor or the marginal soils. Our high throughput phenotypic systems captured efficiently between these variability of these tiller formations and canopy development as well as. In addition, we have also tested another this low soil phosphorus fertility for this lysimetric another phenotyping platforms to see the plant growth and development as well as the agronomic aspects, some of the water use components too. Then this is very clearly like resulted in that. So if you see the graph, in the blue bars are high fertilized phosphorus conditions or optimum phosphorus conditions. These yellow bars in the figure are the low soil phosphorus conditions. We see that greater variability in the number of tillers. If you see the bottom, the low phosphorus ones, there is no any tillers. There is only one main plant will be there. And if high soil phosphorus condition, we could see a lot of tillers here. So this is very similar to the previous ones. The genotype that are adapted to low soil phosphorus conditions that produce more tillers. The more tillers, there is high chances of sustain the crop productions. They will get some of the grain yield in the things. So that's one of the important things we can able to capture very nicely using our phenotyping platforms. The next one. So finally, so we do all this phenotyping in a different phenotyping platform, in a different abiotic stresses and in different conditions. Finally, all these phenotyping data were linked with genotyping data in order to identify genomic regions that are responsible for abiotic stress adaptations. Here I didn't put more slides on this genomic regions for the physiological traits. I just compressed. There are a few things. Yeah, we have also mapped a lot of QTLs and marker trait associations for some of the physiological traits here. I would say that canopy development traits, especially canopy architecture, tillers, plant height, leaf area, those things. For water-related traits, for transpiration rate, transpiration efficiency, all these traits, we have identified QTLs in a different genetic backgrounds, and we have markers too. And also some of the crop production related traits. Especially for the panicle harvest indexed, grain number, and a grain yield. In case of pearl millet and chickpea, we found that water use related traits were co-located with crop production related traits. which are previously reported for drought adaptations. That's here also we showcased here. In summary, the utilization of high throughput precision phenotyping technology hold a great potential in various areas. It allows for efficient evaluation of germplasm under abiotic stress conditions, that leading to better understanding of genotype by environment interactions. Additionally, these high throughput phenotyping platforms enable us for mapping of quantitative trait loci or marker trait associations in a specific component traits in a given environment conditions. This technology plays really crucial role in breeding efforts aimed at developing climate-ready cultivar that can survive for diverse environmental conditions. Thank you so much for teams at crop physiology and genomics and breeding units and gene bank, and many thanks for organizing this training, this lecture organized by several institutions and people. Thank you so much. Yeah, I'm happy now. I finished my presentation. I'm happy to take questions.
रोहित जी चैट बॉक्स ऑन करिए, जो पार्टिसिपेंट्स अपनी क्वेरी पूछना चाहते हैं, इनकी क्वेरी है वो चैट बॉक्स में लिखेंगे और सर उसको रिजॉल्व करेंगे.
Done, sir, already.
Shall I stop share my slides? Shall I take questions, or how I can proceed?
आप प्लीज चैट बॉक्स देखिए सर.
Okay. Yeah. So here, yes, I see that a lot of questions are coming up. So I will answer some of the questions. Sanji was asking how the sensor help for screening the stresses. Yes, the sensor-based technology, that's what I have shown in my presentations. There are different kind of sensors, this 3D imaging sensor, other RGPs, and there are different sensors are available. But I mean, all the sensors can produce all the data sets, but in our team, we have validated each sensor, how the sensor really reliable for any trait adaptations or any traits for the screening. So all those things we evaluated, we tested, we have presented, we published many papers using this aspect. Please go through our papers, you will get more information. And how the technology reached to field or farmers. Yes, some of our whatever the technology or whatever this high throughput platforms that showed, that is very close to the field, because we always maintain to this field-based densities and field crops. That's much more closer to the field. So we already validated, we have a data and presented in the things. Even some of farmer participate approach also, we showcased our facility, that can be used for this to select some of the lines that screened in these facilities. One question here, what is the difference between diversity panel and cultivar? Yeah, diversity panel is simply the germplasms, that is pure germplasm lines. The cultivars are that has improved all agronomy practices. But this diversity panels for agronomically, some lines are superior, but not most of them. But there's some of the specific traits, diversity panels are really the rich sources. For the cultivars, that's it. Cultivars are already exist in the farmer's field. We call it as a cultivars. That's what the difference between that. Which device we can use to identify the nutrient deficiency in plant available for market easily for small farmers? Nutrient deficiencies. Yeah, I think for this questions, yeah, there are some smart tools are available. So some of handheld tools, sensor based tools are there. Maybe yeah, if needed, we will discuss more on this. Maybe yeah, I will say that maybe some of the NIS based or the mobile sensor based some technologies tools are there. I will, if needed, I will share it a bit later. This sensor working on remote sensing technology. Yeah, this is sensor for 3D cloud-based sensor technology. This is something, it's a laser, 3D laser technologies. That's, if you see this 3D laser scanning, any laser projected on the object, it will reflect, that can be captured as images. From that, a lot of features are extracted, all the traits can be extracted. Yeah, really good questions. How evapotranspiration is regulated in drought adaptation? Good. So maybe I didn't talk much more in that. So in this lysimeter facility, so we have this PVC tubes. In that PVC tubes, we do some sort of process of packing of cylinders. Around 35 days of the growth, then we saturate the cylinders. After that, we do packing. Packing in a sense, we keep a small plastic sheet around a lot of plastic beads. This will prevent around 95% of direct soil evaporation. So whatever water loss happened through plant stomata, that kind of called as a plant transpirations. Yeah, we have set of things to nicely remove this evapotranspiration effect. Sorry, soil evaporation directly we can prevent it with that kind of tools. Yeah, and also these days there are a lot of other equation based. We can also evaluate this Penman equation, so we can remove this nicely soil operations. What is the difference between cultivars and variety? I think for me, both of them same. I couldn't see anything different. So yeah, I think more or less similar meanings.
I think no more question.
Yeah.
Hello?
Yes, I
Cultivar,
think.
cultivar is purposely created by humans to enhance choosing traits through artificial selection. But the variety is a version of the plant species that occurs naturally through natural selection.
Thank you, sir. Thank you so much. Yeah, maybe that may be a perfect apt one. Yeah, through these modern technologies, how much time to take breeder to develop a bite-resistant variety? Yes. Now, recently, this rapid generation advancement, this RGA facility will be there. From that, some of our recent things in Chickpea, RGA, rapid generation advancement technology, they come together 3 years, 3 years, they could able to develop one particular things. I want to able to make a cross to, fixing the lines. So in like in 3 years, they will stabilize it. That's what they showed. Which is best, which method is better for transpiration estimation? Yeah, still there. Yeah, there are different tools are available for measuring the plant transpirations. It depends. It depends upon how much material you're going to screen, how much intensity you want to screen. For example, you want to see only one particular variety or one particular genotype to purely understand this photosynthesis and plant transpirations in a diurnal things. Yeah, there are some of our tools are available in this IRCA and porometers. But if you really want to see the different materials or the large number of genotypes, maybe that particular technique is not suitable. You have to go for this either high throughput load cell based technology or this manual technologies. Still, this part based weighing method also still prefer. Lysimeter or modified Penman method. Beyond that, because in the lysimeter, yeah, there is some similarity, but lysimeters beyond that, we can very precisely quantify the water use parameters as well as agronomy. That's one of the major thing that will really provide this facility. Also, that can be prevented in the rain and other things. The other questions, what is the potential of drought adaptation in case of normal season? Yeah, when we look at the drought, so there is two things, one for the soil drought, another one is atmospheric drought. If any plants or if any variety or any material, even under well water conditions, they can able to manage water efficiently, surely that particular genotype can adapt even under drought stress conditions or the normal phenomenon. I would say that. How does phosphorus will affect the tillering process? Yes, really greatly affecting this tillering process, especially in a foxtail millet. Because if producing more biomass, surely plant need this fertilizations, these fertilizer things, this organic and inorganic. I mean, for the organic fertilizer things, especially for the NPK. So phosphorus is the major key for plant growth and development. Surely the tillering, one of the key things, will affected by low phosphorus. How this technology work among with spectral light? Yeah, this sensor now we have updated for RGB and RGB plus as well as NIRS things. Still, the spectra will work for both day and night things. It's the same technology which I was doing with IRI. IRI has the technology that technology provided by Lamna Tech that has conveyor belt systems where plant moves through conveyor belt. There is one stationary point of imaging cabinet. From that, plant slowly move to the conveyor belt to the imaging cabinet. From there, they're taking images. For this technology is a bit different. Here, plant is not moving. Plant is sitting on one particular position, one particular place. This camera and sensor is moving. This camera is moving, or this whole sensor is moving towards a plant to taking the images. That's really two different, sensor technologies. One, this is Lamna Tech. These are some of the Phenotyping. They have two different service provider. Really different technologies. Are you using this technology greenhouse? No, we are using this technology in the open field, open environment conditions, kind of open, because at the end, we're going to use this, whatever the information derived from this facility, we're going to link with open field based phenotyping information. Yes, our facility is there in the open field. It will face rain, it will face heat, everything, it can be normally, it can face.
I think, sir, no more question in the chat box.
Yeah, almost I'm done. I think.
Sir. Thank you so much for joining us, and looking forward your support in the future academic activities organized by our university and foundation. If possible, then kindly share the PPT, sir. Several participants are asking for the PPT.
Sure.
I'm writing my email in the chat box. You are kindly requested to please send the PPT. I'm writing my email, sir.
Sure, sir.
Ankur@csgmu.ac.in. This is my official email ID.
Sure, sir. I will take a photo. Then I will. So in our team, we have this series of presentations are happening. So I think we have discussed internally. We will going to share all the.
No issue, sir. I know, sir. There are some IPR issues in your
No,
institute.
not. I mean, not for that. We thought, we're going to put everything, one particular things. We're going to share it.
Okay.
That's
Got
best
it, sir.
platform. Instead of I'm sharing alone, and again other colleagues also sharing a lot of things. Surely we will share, sir. No issue.
Thank
This
you.
is
Thank you,
open.
sir.
Open.
Thank you for acknowledging my point, sir. Thank you.
Yep. Thank you so much, sir.
Okay,
This
sir.
is
Good
really
night.
great opportunity for me, to present, to share
Sure,
the
sir.
experiences in a different kind of audience. Yeah, it's really broad and great, and you guys are taking great initiative for teaching or giving this opportunity for all the students or all the faculties, all the people around. This is really important for at this stage.
Thank you, sir, for your positive word of mouth.
Okay, I think really great evening. Yes, surely I'm happy to contribute in future like this kind of events.
Okay, thank you.
Thank you so much for everyone to attend this presentation, this conference. Okay, I'm stopping things.
रोहित जी रिकॉर्डिंग ऑफ करके